Good morning, good afternoon, everybody. Welcome to today's webinar. So my name's John Brown from Bristol Myers Squibb, and I'll be chairing today's session on behalf of the GastroPlus user group. So today's webinar has the title of Integrating Mechanistic in Vitro Dissolution and PBPK Models to Drive Drug Development. And we're fortunate today to have two speakers. We have Jim Mullin and Sumit Basu. They're hosting approximately half of the webinar each. We will be taking questions, but we'll take all the questions at the end. So if you have a question that you'd like to submit, please type it into the question box in the software, and then we can raise those questions after both speakers have given their presentations. As for previous webinars, we'll be recording the webinar and it will be available on the GastroPlus user group website if you want to go back and listen again or share it with colleagues. Okay, so without further ado, I'm going to introduce Jim Mullin. Jim is currently a team leader at Simulations Plus. He has 9 years experience in pharmaceuticals, where he's focused on process modeling and computational fluid dynamics. He's a chemical engineer by training, and prior to Simulations Plus, he worked at Bend Research. And today, Jim's going to talk about how we can use GastroPlus in vivo simulations and link those with DDDPlus simulations of the in vitro situation. So, Jim, if you'd like to go ahead and start, it'd be great to hear from you.
Yes, thank you. I'll dispense with the title slide after that introduction, and we'll get right into the meat of the presentation. So today I'm going to talk about the utilization of IV IVC and dissolution modeling to design a formulation to hit a target PK profile. This uses the combination of the IV IVC functionality in GastroPlus and the dissolution modeling capabilities of DDDPlus together in performing this type of modeling. Also, we'll show how you can use the simulated dissolution variability features or sort of population simulations within DDDPlus to look at the variability in your in vitro experiment and see what effect that has on your BE within GastroPlus using the population simulator. Then second, I'll look at the biphasic dissolution test. This is one of the new models we're adding to the next version of DDDPlus, where you can predict the drug diffusing into the organic layer. And the goal with this model is to extrapolate precipitation kinetics where there's an absorptive phase in the dissolution experiment. And so we'll go through a little bit of the model development and a case study for Intra Kanazawa. So how can the dissolution software be used, DDDPlus in particular? One such way it can be used is in dissolution method development. So how can I design my in vitro experiment to correlate with my in vivo release? So of course, in GastroPlus, and you know that there's changes in pH as you go from the stomach to the small intestine. There's changes in volume. There's changes in bile salt content. So the solubility changes, with time, and etc. In your in vitro experiment, you can do multi-stage dissolution tests. You can choose the media that you want, the volume, etc. And with the IV IVC features in GastroPlus and knowing the true in vivo dissolution rate or being able to deconvolute that, you can compare your in vitro situation directly with what your in vivo release profile is. And you can see how you can maybe change your in vitro experiment to more one-to-one correlate with the in vivo release. In formulation design, you can also ask questions like, how do I modify the formulation? How much polymer do I need in a CR matrix tablet to impact the dissolution rate that that will meet my target in vivo dissolution profile, and then PK. So we'll be talking about mainly the formulation design today. But those are two applications of both the softwares together. And then the last application we'll be talking about today is, sort of establishing dissolution specifications using the DDDPlus simulation tool. Once you have a model built for dissolution, you can look at the variability of any of the parameters in your dissolution experiment and see what effect that has on the dissolution rate or the family of dissolution rates that you would expect based on these variabilities. So you can really look at both the high and low dissolution curves that are predicted based on, let's say, changing polymer amount in your tablet, maybe the variability in the pH of your experiment, other variabilities that may exist. And once you have this, population of dissolution curves, you can see or simulate what effect that has in vivo using the GastroPlus ACAP model and PBPK simulations, and see what effect that's going to have on the exposure. You can also run virtual BE trials, which we'll show in the example later, so I'm not going to harp on this slide too much. We'll get into this in more detail. So this is a little bit of a busy slide, but this goes through the overarching steps that you're going to have to perform to do model-based formulation design. So the first step is you're going to have to know how your in vitro dissolution rate is going to impact your in vivo dissolution rate and resulting PK profiles. So to do that, you're going to have to use GastroPlus and generate an IV IVC using the mechanistic-based IV IVC tools. This will allow you to get a correlation between the fraction of in vitro release in your in vitro experiment versus the y-axis here, which is actually the fraction in vivo release. So once you have that correlation, you directly know how your in vitro experiment is going to be relating to your in vivo release. So once you know that, you can then define a target PK profile that you want to hit. And if you're a generic company, this would be an innovator product, right? Some product that you're trying to match bioequivalence with. And so you need to define that profile. Once you define that profile, you can use GastroPlus to deconvolute the in vivo dissolution rate that will produce the PK profile that is your target. So in the step 3 here, it shows that target PK profile. We use the Weibull function here, which is this blue curve, to determine the in vivo dissolution rate, which directly matches that PK curve. So we know the true in vivo dissolution rate that will hit that target. Once we know this true in vivo dissolution rate, we can use the IV IVC generated in step 1, and then we can convert that to an in vitro dissolution rate. So in step 4, if you use that IV IVC relationship, you can convert your in vivo dissolution target to a corresponding in vitro profile. And once you know that in vitro profile, then you have a target with which you can try to do some formulation development, right? So you know in your in vitro experiment what exact target I need to hit. You can build a model in DDDPlus for the release of your tablets, your CR tablets in this case, and try to determine a formulation that will match the target in vitro profile, which is represented by the red square points in this graph on the bottom right here. So that's the overall arching goal of how you link back the in vivo situation from GastroPlus to the in vitro situation in DDDPlus, and how you might affect formulation development using these two tools. So what is an in vitro to in vivo correlation? The working definition is that it's a predictive mathematical treatment describing the relationship between an in vitro property. In this case, it's going to be the in vitro dissolution rate and a relevant in vivo response. Usually that's the plasma concentration time data if you're using Wagner-Nelson or Lou-Riggleman deconvolution. But within GastroPlus, we have the mechanistic-based IV IVC that goes one step further and can actually use the ACAP model to deconvolute the in vivo dissolution rate not just the systemic concentrations. So in traditional IV IVC, you're simply looking at your in vitro dissolution rate versus the systemic bioavailability. And then there's a big, gray box in between. You don't really know what's going on. You don't know anything about the dissolution rate in vivo. You don't know anything about the permeability, the first pass extraction in the gut or the liver. None of these processes are, enlightened, I guess. Once you use the GastroPlus model, you can use the physics of the model to then go from systemic bioavailability back to the actual drug dissolving in the lumen. So based on the absorption through the enterocytes, any metabolism that occurs, if you could also have active transport processes, that's all taken into account in the ACAT model, along with the transit times, the pHs, the bile salts, etc. Once the drug reaches the portal vein, then it can, go through the liver and be extracted through first pass metabolism. So we really understand the fraction absorbed, the fraction of dose in the portal vein, and then the systemic bioavailability, all to back calculate what the true in vivo dissolution rate is. So this is the mechanistic version of the deconvolution that we perform in GastroPlus. So the example I'll give here is sodium valproate, which comes from this Dutta paper. This data is from 2005. There's 5 different formulations from fast to slow. You can see the PK data here on the left-hand side. There's an IV arm to the study as well, which is the black curve, which is what we use to develop our PBPK model and our systemic clearance for this compound. So the questions were, we want to use this data to show, the utility of maybe having a minimal set of data to try to do some formulation development. So what we did is took a subset of this data, only a couple of the formulations, to simulate what, a company in development might have. They might have limited data. So what we did was build an IV IVC for only 2 of the formulations, the slow and the slightly fast formulation. So the slow is the sort of magenta formulation here, and then the slightly fast formulation is the blue formulation. And then we wanted to see if we could design a tablet formulation that would hit the green PK curve in the study. So that's what we're doing. Can we utilize IV IVC and the in vivo dissolution rate for the fast formulation to predict the in vitro dissolution rate and formulation composition for this green fast formulation for sodium valproate? So the first step 1 in that whole mechanistic outline of how we're going to go about doing this is generating the IV IVC. So as I said, we use the slow and the slightly fast formulation. We selected those as the records that we wanted to build the IV IVC with. We selected the mechanistic absorption model deconvolution method, which is going to give us the information regarding the in vivo dissolution rate. We deconvoluted and then formed a correlation between those. So you can see here it's a second order polynomial function. And then we got this sort of profile for our IV IVC where the x-axis is the fraction in vitro release, and then the y-axis is the in vivo dissolution in the lumen. So now we have a formula that's going to relate our in vitro and in vivo dissolution and have a link to our resulting PK. So step 2 is defining our target PK profile, and in this case we're using the human CR fast profile as our target profile that we wanted to design a dosage form for and hit that as our target. So we selected that in step 2 as our target PK profile. We used GastroPlus in step 3 to extract the in vivo dissolution rate for that formulation, which is the graph here on the far right is the in vivo dissolution rate that results in that plasma concentration prediction based on our PBPK model. Once we had that, we could then use the IV-IVC relationship. We can invert it so that we had a formula between X, the in vitro dissolution, and Y, the in vivo dissolution. So this is the in vivo dissolution. We plug that into this formula, and then we can get X, which is our in vitro dissolution. Those are the red data points. These are at the same sampling time points as their in vitro experiment are, and they're generated from this, this in vivo dissolution rate. So now we have an in vitro profile that's our target, and can we design a formulation to hit that in vitro target? So first, to do that, we're going to need a dissolution model, right? And so we use DDDPlus. But first, let's talk about the actual formulation itself. So we're going to use the slightly fast formulation because it's part of the sort of initial data set that we use to build the IV IVC. So it's a known, we have dissolution data for that, and it's a known formulation. So we can build the dissolution model with the slightly fast formulation, and then what we're going to try to do is predict the amount of HPMC K15M and the filler lubricant, which is lactose in this case, that would give us that target PK profile. So to build the model, we use the Korsemeyer-Pepys equation. Of course, in the limit of the power law constant n being 0.5, this would be the Higuchi equation, the classical square root of time functionality of drug release from a matrix. And that's a theoretical equation. Now, the Korsemeyer-Pepys is a fitted equation, and it's empirical. There's some calibration constants that allow you to take into account the excipients and calibrate it to your complex formulations. So each polymer has its own polymer drug calibration constant, and each polymer dissolves based on its molecular weight, chain radius, and its chain disentanglement concentration. The porosity is updated based on the amount of filler, lubricant, drug, and polymer dissolved, and so the porosity changes over time. And we can calculate the drug release, but we need to calibrate the power law constant and the calibration constant for HPMC in this case to our slightly fast formulation, so that we can build a model that we can then try to predict the fast formulation. So we did that in the next slide. We fit the power law constant and calibration constant for the slightly fast formulation. We obtained a release exponent of about 0.78, which is very normal for swellable matrix formulations. Typically for these types of matrix formulations, you will get a power law constant between 0.5 and 1, which indicates that it is swelling and it's a swellable HPMC matrix formulation. So once we had this model, we could then build a model for the fast formulation, and what we did was create various different tablets for the fast formulation with different HPMC to lactose ratios to match the full tablet weight of 1 gram. So we started at 300 mgs and we decreased, and we looked at the F2 statistic, and we found that 16% HPMC was the optimal formulation polymer composition to hit that target in vitro profile that we determined from the GastroPlus deconvolution and IV IVC. So that's the formulation composition we would have predicted, and then we would have recommended be produced and tested in vivo to try to hit that target PK profile. Of course, is that correct? Well, the actual fast formulation was 20% methicel. So, we were within 20% of the prediction of HPMC. So we got, we had a shot on goal and we're somewhat close. We're not perfect, but it's better than a shot in the dark. We're using the mechanistic model to try to really look at the release and the effect of the different excipients, and really try to go in with more knowledge to our bio study as far as the formulation and trying to hit a target profile. Once you have that dissolution profile and your IV IVC, so once you know the relationship between your in vitro dissolution test and your plasma concentration profile using the GastroPlus model and IV IVC, you can actually look at, there's some more powerful features that you can look at, sort of the variability in your dissolution test. So you can simulate dissolution variability by doing a population simulation within DDDPlus, basically adding variability to the content uniformity of the tablet and some of the parameters in the dissolution experiment. And then you can simulate a family of curves. This is, I think, 25 virtual trials, and it's showing the probability contours of those dissolution experiments, simulated dissolution experiments. From that, you can get sort of the range you might expect from your in vitro dissolution. And since you have an IV IVC that will directly translate this into the in vivo dissolution rate and plasma concentration, you can test whether or not the high and low ranges are bioequivalent. So in this case, we ran the test and reference as the high and low dissolution formulation within GastroPlus using the population simulator, and we got the fact that these are bioequivalent. So based on some fairly pessimistic CVs that we used for content uniformity, you would feel that you're pretty safe in this case in passing bioequivalence based on the population simulations. Okay, so the next section of the talk will be looking at the biphasic dissolution experiment and the new model that we built into DDDPlus to look at the simultaneous dissolution and then diffusion of the active into the organic layer. And the goal with this is to have a absorptive dissolution test where precipitation kinetics can be extracted. Of course, an absorptive dissolution test is interesting because the absorption part of the experiment will reduce supersaturation and change the precipitation rates. And using these types of methods, we may be able to extract better precipitation kinetics from our in vitro tests. So the biphasic dissolution model is shown here in the slide 18. There's an aqueous layer, an organic layer, and then there's diffusion through the stagnant boundary layer between the two phases. The flux is calculated as shown, and if you rearrange these equations and solve, you can get a differential equation for the appearance of drug in the organic layer versus time. And that's strongly related to the octanol water partition coefficient or decanol or whatever organic you use, phase you use, and the mass transfer coefficients. The diffusion coefficients we can predict from structure. However, the boundary layer thicknesses are not really very well known in these systems, and so you have to maybe fit these. Of course, there's other literature that have looked at these values, and they're somewhere around 30 micron in thickness. So we utilized biphasic data from this study from Carl Box at Sirius Analytical, which is now Pion. They did this biphasic dissolution with various compounds that are ionized and neutral at physiologic pH of 6.5. These were all done in phosphate buffer, and these are all solution doses. So based on the solubility at pH 6.5, there'll either be precipitation or no precipitation. So I think the compounds on the left there was very little, if no, precipitation in any of those based on the level of supersaturation and or propensity to crystallize. And then on the right-hand side, there's some neutral compounds and ionizable compounds where there is supersaturation at pH 6.5 and there is precipitation. So the experimental conditions are shown there on the bottom left. They're small volume experiments, 40 mils aqueous, 30 mils organic. These are decanol and 5 mg dose for each of these. So I'm only going to look at a subset of this data because we don't have time to look into all of them. I'm just going to look at a couple of the compounds that don't precipitate. So first, Indomethacin, which is a lipophilic compound. It's an acid, and it's got a pK of around 3 or 4. So it's going to be soluble at pH 6.5. There's no supersaturation, so we're looking purely at diffusion into the organic layer and using the experimental aqueous solubility and then the organic solubility predicted from the log D versus pH profile. we use those as inputs. We didn't fit them. We just fit the aqueous boundary layers. We got somewhere around 20 microns for the aqueous boundary layer, which is very similar to literature. I think the paper from Michigan on the biphasic dissolution that came out quite a while ago from Deanna Moody. I think those were in line with what she had in her paper. So we're pretty convinced our model is reasonable. Valsartan, again, a lipophilic compound. Again, it's going to be, in this case, more soluble at pH 6.5 in the aqueous than the organics. And so what you see based on the experimental solubilities and the prediction of organic solubility based on log D versus pH, we see that we predict the plateau of the dissolution experiment fairly well just with the experimental solubility data. And again, our boundary layer is around 30 to 40 micron in this case. So we were pretty convinced that our model was, accurately portraying the simplest case of all when you're dosing a solution to the biphasic dissolution experiment. So next we looked at one where it's going to precipitate and it's relevant to the in vivo situation. And so to do this, we're going to use the mechanistic nucleation and growth precipitation model. The nucleation rate is going to be calculated based on the equation shown above, and so we sort of lump this into the pre-exponential and exponential term, but it's really all just a mathematical representation of the free energy change going from a supersaturated solution to a nuclei of a given critical radius. Of course, there's the frequency of molecular collisions based on the level of supersaturation and whatnot. The key parameters here are the interfacial tension of the compound, which is going to determine, sort of the free energy of the critical radius and whatnot. So that equation is going to be used to calculate the nucleation rate. As nuclei are formed, the crystal can grow. And keep in mind, this is all homogeneous nucleation. And we have a few fitted constants in there to match experimental data. So there's the exponential correction factor and the Lin-Force parameter where we can adjust the precipitation or nucleation rate to match in vitro data. The crystal growth is basically the same equation as Noyes-Whitney, but in reverse. Instead of dissolution away from the particle, you're going to have drug from the bulk diffusing to the surface of the crystal so that it can grow. So that's the diffusional crystal growth mechanism is what we're assuming currently. So for itraconazole, a very high log P compound of about 5.6 or so, it's got some basic PKs, so it's more soluble in the gastric environment than the intestinal environment. So there's a good deal of a driving force for precipitation in vivo. So we wanted to use the in vitro experiment from the literature from Carl's dissolution experiment to see if we could predict in vivo precipitation using our GastroPlus model. And so we built this DDDPlus model. We extracted the precipitation parameters. The left-hand graph is first-order precipitation. The precipitation time was about 302 seconds. The precipitation size is calculated sort of based on the slope of the organic curve after the drug is precipitated. So you can see from about 15 minutes to 60 minutes, the organic is slowly increasing in concentration, and that's determined by the redissolution from the extremely low solubility and small particle size of itraconazole. The mechanistic nucleation model, we extracted the exponential correction factor and then the Lin-Force parameter to fit the in vitro data. And the precipitation size, again, is somewhere less than a micron, so very small radius of precipitate is predicted based on these models. So the goal was, can we use these parameters that we extracted from DDDPlus, can we use them directly within GastroPlus and predict the in vivo situation? So my colleague Kay Jetto from Simulations Plus developed a itraconazole PBPK model for the drug-drug interaction with midazolam. And so she looked at 9 different formulations, co-dosed with midazolam and looking at the DDI. We're going to simply look at the formulations where they did day 1 dosing with no no interacting compound. So, but we're going to take her model. So if you want to know the details of the model, I would suggest looking at this poster. We don't have time to go through the development of the whole model, but I'm using her exact model from this work that she'd presented at APS to look at the precipitation parameters and see if they work in vivo. So first off, on the left-hand side for the next 3 graphs, or 3 or 4 slides, are going to be the predictions in GastroPlus, where we use the in vitro dissolution precipitation parameters. And then the right-hand side graph is going to be the precipitation parameters that were fit across all those 9 in vivo experiments from Kay's poster. So keep that in mind. The right-hand side is going to be predictions based on the precipitation fits to a global data set from her work, and then the left-hand side is going to be from our DDDPlus simulation. So for this fed 200 mg tablet, we don't really see much precipitation. Obviously, the pH is a little bit higher in the stomach in fed state, and there's more bile salt, so we don't really see much precipitation in the fed state for the 200 mg. In the fasted state, we do see some precipitation, and there is some differences between the capsule for the 200 mg dose of Vitro Kanazawa. On the left, you can see the Cmax is decreased, about 10 or 15. Well, maybe not that, but probably only about 10 or so nanograms per mil on the left-hand side using the in vitro parameters versus the right-hand side, which is the fitted parameters to the global data set. Again, for the fed state, this is a PO solution dose of 200 mg itraconazole. We predict a little bit higher precipitation with the parameters from DDDPlus. As you can see, the red curve is the amount dissolved, and you can see it drops to a much lower value here initially, which shows a little bit higher precipitation using the parameters from the in vitro experiment. But overall, the redissolution and whatnot, we get a pretty similar profile for the fed state for the PO solution. So in this case, the in vitro parameters work pretty well. The final case is the PO solution in the fasted state, and this we again predict more precipitation, but since we're in the fasted state and there's not as many bile salts and the pH is lower in the stomach and whatnot, we're just not getting the redissolution as much back up to to match the PK curve. So in this case, our parameters from the in vitro experiment over-predicted precipitation, and we've seen this if you've come to these meetings for the last 2 or 3 years. I think in the past we've presented on, for the most part, in vitro precipitation assays generally overpredict in vivo precipitation, and that's true of, I think if you look into some of the work from Sarah Carlert, from the AstraZeneca compounds, and then dipyridamole is another one where in vitro you predict a lot of precipitation, in vivo you just don't see it, you see very moderate amounts. And we're seeing this in this case for the biphasic dissolution test as well. It looks a little bit better. We're predicting some of the formulations like the fed solution fairly well, but in the end we overpredict precipitation. So in summary, IV IVC and dissolution modeling can provide a framework to design dosage forms and maybe reduce studies for new prototype formulations. And the biphasic model, you can look at precipitation with an absorptive phase to look and see if a reduction in the supersaturation based on the absorption or partitioning into the organic phase improve the precipitation predictions. We're currently developing membrane permeation and artificial stomach duodenum to that end to try to extract better precipitation parameters for our in vivo simulations. So I think that's it. Acknowledge all of the simulation team here, and then of course Mike Bolger, our chief scientist, and I'll turn it over to.
Okay, thank you very much, Jim.
Yep.
So if you have any questions for Jim, please put them up on the question box, and we'll come back to them after our second presentation, which is along very similar lines. This time it's from Sumit Basu, who currently works for AbbVie, where he supports projects across therapeutic areas of mechanistic modeling. Sumit received his undergraduate master's degree in pharmaceutics from Jadavpur University in India, and he then went on to complete a PhD at the University of Houston in pharmaceutics and postdoctoral training in the University of Florida. So Sumit's here, and he's going to talk to us about how a PBPK model can be used to simulate formulation differences in ER formulation work. So Sumit, over to you.
Okay. Thank you, John. I just would like to make sure that my slides are visible. Can you see my slides?
Yes, we can.
Okay, thank you. Yeah. So thanks Simulation Plus first of all to give me an opportunity to present my work. This work actually I have done in University of Florida, and basically it's a big project from FDA, from Office of Generic Drug with University of Florida, and I am just presenting one particular case study how we actually used GastroPlus as well as DDDPlus to address different clinically relevant questions. So just to begin, I would just give a perspective that how what is the problem and what are the challenges and how we are addressing those particular questions. So we already know that in US, the utilization of generic drugs actually accelerated a lot in last 10 years, which is actually kind of beneficial for the US health system. And for generic drugs, basically these are considered safe and effective since they always undergo a rigorous bioequivalence testing. But the problem is that some patients, and this is like a good patient population, they always report that when they're switching from brand to generic drug, or sometimes some other generic drug to second generic drug, they are experiencing different type of adverse effects. Also, sometimes there is a kind of lack of efficacy for those particular generic formulations. So OGD actually wanted to see that if there is any mechanism behind why this kind of adverse effects are causing, so that's why they wanted to use a modeling simulation approach. And in University of Florida, we are actually using 3 different modeling and simulation approach to address this kind of questions. So basically for anti-epileptic drugs, it's well known that there might be a purported therapeutic inequivalence whenever you are switching from brand drug to the generic drug. And also it has been predicted that for the new compounds like novel oral anticoagulants, sometimes it might happen that since they are like narrow therapeutic index in nature, so from whenever they're switching from the brand drug to generic drug, there might be a potential risk of bioinequivalence. So that's why we are trying to answer these questions, that whether there is any kind of drug effect or system effect or any kind of formulation effect is behind to change this kind of bioinequivalence. We used a modeling and simulation approach where we are using 3 different integration approaches, factors. Like first is PBPK model, where we'll be seeing that how or what are the reasons, like physiological or drug or system or formulation is causing any kind of bioinequivalence. Then we'll be using that PK simulation to our PKPD model platform and determine that whether that change of PK is really causing any kind of PD change. And thirdly, we'll be using a bioinformatic tools to determine that the patients, whatever they are actually showing, the experiencing the pain, whether this is true or this is just a noise, whether we can differentiate it noise from the signal or not. So this particular study, I will just show that for metoprolol, which is kind of representative of extended release formulation here, how we can address different clinical relevant questions by evaluating impact of the formulation factors on in silico bioequivalence. Just 1 minute, I think it freezed. So I just would like to show you that why we actually took metoprolol as an example, because we actually got some of the reports, and also we received from US FDA that they actually received like over 3,000 adverse incident reports for drugs, which actually showing that lack of effectiveness, as well as there are some troublesome side effects. And also from different articles or different news, they actually showed that whenever patients are patients are taking generic metoprolol, sometimes they're experiencing chest pain. And even drug companies, generic drug companies, they are sometimes like changing or like discarding the generic drug bottles because of the dissolution test failure. So I will show that why what are the conventional metrics is changing. That's why the dissolution test is failure. So FDA actually have a stringent policy, and they already published that in 2015. Back in 2015, that most of the time that whenever you are actually manufacturing a generic drug, so first you have to evaluate the or you have to perform a bioequivalence study, and for that particular bioequivalence study, you have to calculate the 90% confidence interval for ratios of geometric means of AUC and Cmax of brand and generic. And if it is lying between 80 to 125%, then only you can actually market your drug. So one thing is there for generic company that once you pass this test, and once your drug is in the market, so there is no kind of quality control check that whether your other batches or other lots are actually similar. And second thing is that for basically for BCS class 1 drugs, since we know that they have high solubility and high permeability, so from in vitro dissolution study, you can actually calculate both indices like F1 and F2, which is known as more similarity and difference metrics. And if you can show that your F2 value is greater than 50, greater than equal to 50, then you can actually waive your BE test or option A. So Jim actually already showed that how you can calculate the F1 and F2. So without wasting time, I'm just going to the next slide. Just I would like to point out that this F1 and F2 system is only applicable for BCS class 1, and recently it has also been applicable for BCS class 2, provided the excipients what you are using in brand and generic are qualitatively same and quantitatively also same. But these F1 and F2 tools are actually empirical in nature. So whenever you are designing a formulation, you don't know that which particular mechanism might actually change the PK performance of your drug. So that's why you can actually use modeling simulation approach. And here I provided that how we can actually make a conceptual framework to use or to determine what are the important factors is actually impacting on your PK profile of the metoprolol ER. So suppose we have a formulation and then it's disintegrating into the dissolution medium. So we can easily can use DDDPlus to actually determine or to develop an in silico dissolution profile, and from there we can link with our absorption model where we are actually keeping the disposition kinetics constant, and after that we can actually simulate the PK profile with this particular modeling, and can, without doing any BE test, we can see whether our AUC and Cmax is within the acceptable BE range or not. So I will describe this figure more in later slides. Before that, let me introduce that how we actually developed for absorption model, and then how we actually linked with these with the formulation factors. So basically, for PBPK model, we have actually a nice article where they are giving IV infusion, and we use that particular infusion data to determine what is the disposition kinetics of metoprolol. So using PK plus of GastroPlus, we actually see the main structural model is 2-compartment PK model, and you can see in the right side that our predicted concentration is in good agreement with the observed concentration for 5 mg IV infusion. And then using the same disposition kinetics, we actually validated our model with 10 mg IV infusion, and we found out that which actually increased the confidence of our PBPK model. So just like I would like to mention, the nature of this model is more like mechanistic absorption model. We are not putting any kind of enzyme and transporters in the disposition, because we know that metoprolol is metabolized by CYP2D6, but since we are more interested in the dissolution and absorption performance, so that's why we are using kind of like hybrid model where we're defining the PK with compartmental approach, and then we are checking the absorption and dissolution with the help of the ACAT model. So in our second step, once we define our disposition characteristics of the API, then we went to calculate went to investigate for the absorption model, and which is kind of coming from the immediate release formulation of metoprolol. Since metoprolol doesn't have any kind of dissolution profile for immediate release formulation, that means like whenever you are giving IR formulation, it will be dissolved very quickly in the body. So that's why we didn't change any default ACAT model. We just introduced the physiochemical properties of the API like metoprolol, and also the other kinetic factors like phosphorus metabolism, phosphorus extraction, or the intestinal factors in our second model. And this in stepwise fashion, when we introduced this complexity, we calculated this one. We actually developed this model in 50 milligram immediate release formulation. And what we found out that our Cmax and AUC with PBPK model prediction is in line with the observed data. And then again, we validated with this particular model with another dose like 100 milligram first release, where we found out again it's in good agreement with the observed data. So once we are actually sure about the immediate release formulation, IR absorption model, then we wanted to develop the ER formulation or ER absorption model. But since for ER formulation, there is evidence from the literature that metoprolol might undergo a sufficient colonic absorption, so that's why we wanted to check with a colonic metoprolol formulation that what is the maximum absorption capacity for metoprolol. So we utilize that data where metoprolol is only given as a colonic perfusion in the colon, and in our model we use the transit time for stomach intestine as a 0 minute, and then we calculate the absorption scaling factor, which is there in the ACAT, which actually introduces that what is the segmental distribution of different the segmented distribution of different enzyme and transporters. So that's giving us kind of feature that how the drug is absorbed in different segment of intestine. So basically, once we get the colonic ASF values, we integrated with our ACAT model and optimized it, and then we developed our ER absorption model. So in the last case, you can see that we are actually stepwise building all three different factors in ER absorption model, because in IV infusion, we are first taking account of the disposition. In the IR absorption model, we are adding more absorption plus disposition and physical chemical properties. And lastly, with top of that, we are actually adding our in vitro dissolution profile of that particular formulation. So since here we wanted to know more about the effect of the dissolution, we didn't use the Weibull parameter, and we knew that for the purpose of our work, we had to see that what are the mechanistic factors can change dissolution. So that's why instead of using Weibull parameter, we just wanted to see that what are the other mechanistic factors using DDDPlus can change the dissolution profile. So here you can see in the step 3 that we had this in vitro dissolution profile from that particular article, and then using that data, we and optimized ACAT model, we can predict the in vivo PK profile here. And not only we predict this one for 25 milligram, we also use that particular model for other dose like 50 milligram and 200 milligram. So when we were seeing that when we got that model developed and validated, we were interested that what particular factor might be responsible to change of absorption in metoprolol. So if you can see from this figure for IR absorption, IR formulation, the absorption site was more on small intestine, but whenever we are switching to the ER formulation, it's actually going to the large intestine. So this is actually giving us a hint that apart from like maybe among all drug system and formulation factors, formulation will be the main important thing which can cause a bioinequivalence when we are switching from brand to generic drugs. So then we systematically conducted different other factors and determine the systematically conducted sensitivity analysis to see the effect of transit time or the formulation type and also the system properties. So what we found out for IR formulation, most of the time whenever you are introducing the drug, it's actually giving you identical result what you are predicting. There is not much sensitivity analysis is going on here, and this is one of the facts because metoprolol have high solubility and high permeability, so we also won't expect that it will change drastically something with Cmax and AUC. And in case of ER formulation, what we found out that if you are lowering the colonic transit time, that means if you are kind of changing your ER formulation to almost like IR formulation, then you might actually change, you might actually get a lower BIN. So that's why, or lower AUC. So that's why it can say that it is totally dictated by the formulation type, which has given us another evidence that formulation is the most important factor when you are switching metoprolol from brand to generic drug. And this is just like an effect of primary permeability we saw in our simulation. So we saw that in lower permeability, the bioavailability is really low, both in AUC and Cmax. But in case of bioequivalence, it doesn't matter because whenever we will be doing the BE study, the population will be remaining the same and it will be in the healthy volunteer. So in this study, we are not including any other like gastric bypass surgery patients and others where the permeability might be severely compromised. So that's why we didn't include primary permeability as a significant factor in our to evaluate the bioequivalence. So after that, we wanted to apply to address our question that okay, our absorption and disposition is now constant. Now let's see that if we change the formulation specific factors, how can it change the dissolution input parameters, and as well as it can change the performance of the formulation. So again, I am introducing that figure just to like show you that how we deconstructed each and every factors from this particular system. So we have the ACAT model, which is actually taking account of the absorption. We have the DDDPlus software, which is actually taking account of the dissolution. And ultimately, we have the formulation, which after disintegrating coming to the dissolution. And we also had the amount of components in our formulation, which is giving us an opportunity to tweak different parameters and see that which particular change of dissolution input or output give us a change of BIM. So basically, we got this particular amount from a toprolexol, which is a reference drug, and the drug is actually given as a ER formulation, and the polymer is used as HPMC. So it's actually coated for easier swallowing. And the release rate of that particular drug from that polymer is governed by the Coursemire kinetics, which is already shown by Jim. I am just using here the very simplified formula, which is the fraction of drug release at time t. It totally depends on your release constant K and release exponent N. So K is basically is giving you that how drug polymer interaction is giving a different type of properties of that particular interaction like porosity and tortuosity, whereas N is giving you the nature of the release, whether it's the erosion or diffusion. So as we already know from the previous talk that if you're N equal to 0.5, then it's actually giving you release, diffusion mediated release, whereas when it is greater than 1, it's actually giving us erosion mechanism, what is like more like a zero order release. So what we found out that in our IR formulation, we got a more like a diffusion nature, and in case of ER formulation, it's actually giving us a erosion type of release. So just to like identify more, because we are actually seeing this from the perspective of formulator, that what is the implication of N and K. So N is basically indicative of the release mechanism, which is mainly governed by type, grade, or molecular weight of the release controlling polymer, in this case HPMC. So what we understood that this particular change of N is fairly reproducible because most of the time we'll be using the identical polymer. But in case of K, which is more like an indicator of release rate from a swellable polymer matrix, it might actually vary lab to lab or batch to batch, because whenever we are using HPMC and it comes in contact with the body fluid, it makes a gel. And from that gel, so that gel performance, it depends on the porosity and the tortuosity of the polymer matrix, which can be highly variable depending on which particular processing condition you are using, like what kind of temperature, what kind of humidity, or what kind of excipients you are using. So depending on that processing condition, your K might be variable, and that might actually change the dissolution profile, which ultimately can cause difference in the PK parameters like AUC and Cmax. So that's why we employed DDDPlus after utilizing the dissolution medium pH, and the speed of the apparatus and what type of apparatus are using, and ultimately the amount of the formulation and component of the properties of the formulation in that particular tablet. And then what we found out that once we developed our model in 25 milligram, we actually extended to other dose like 50 and 200 milligram. So in this figure, you can see that the blue line is actually representing the predicted profile, and the red circles are presenting the observed data. So from once we are confident that our DDDPlus model can actually predict the observed data, then we are changing different type of other formulation variable like N and K to determine that how the shape is how the shape looks like. And so basically it's done for the 3 different dose like 25, 50, and 200 milligram. And after doing that, we wanted to use this as input in our absorption model to determine whether change of N and K how is influencing the PK parameter of metoprolol or not. So before going to that, we just wanted to first check that how change of N and K can change of influence the in vitro B of metoprolol. And what we found out that when N is really sensitive than the K to cause BIN, because you can see that whenever we are changing around 5% or even less than 3 to 4%, then it's actually crossing the 50 barrier. F to less than 50, it becomes F to less than 50, so which is causing actually bioinequivalence. But in case of K, that particular thing happens when we are increasing K up to 25% to 28%. So this is giving us kind of a confidence that N is more sensitive than K. But as we said that you can control N really well because it's more like a highly reproducible and it's just a polymer property. But in case of K, it's kind of a drug polymer interaction, and it can operate in its operation result can vary in different processing conditions. So that's why we had to look into that how K is actually affecting the PK performance. So after inputting that dissolution profile, varying K and N, we put in our absorption model in 3 different doses, and simultaneously we wanted to compare with our FDA recognized F2 value, that how confident we are that we are predicting the same model performance. So you can see in the upper panel that we are increasing K both on AUC and Cmax, and in the lower panel we are decreasing K on AUC and Cmax. And each time that in vitro bioequivalence results is quite comparable with the in vivo bioequivalence result. And again, like K, we also did the same thing with the N, and what we found out that we are actually getting pretty confident result that our in vitro BE result and in vivo BE results are quite comparable. And we already know that metoprolol is a high solubility, high permeability drug, so that's why only change of formulation factor might cause some part of bioinequivalence. So in this regard, if we can increase in more than 10% or 9%, 15%, that might also give us like the different dissolution profile, or as well as can change different PK matrix, which might cause the bioinequivalent. So for the new formulation, what you can do that you can also develop like you have this absorption framework, and then you can just do the dissolution profile. You can conduct that, and then using that particular dissolution profile, you can calculate F1, F2, and just to like gain confidence, you can also use that particular dissolution input in that absorption framework and can see that whether your dissolution input really is changing the PK matrix or not. So which can give you extra confidence, and also you can actually show them using modeling and simulation approach that in that particular range of N and K, you are getting VIN, so you don't have to worry about the change of the dissolution input anymore. So to summarize this, I just would like to mention that here we are developing a conceptual framework to identify drug, system, and formulation specific factors which can actually contribute differences in relative bioavailability between two formulations, like metoprolol you can do in the reference formulation as well as the test formulation. And second thing is that we can identify that here that formulation specific factors are critical factors which should be considered during the evaluation of metoprolol ER formulation, which also supports the fact that the established IV IVC of metoprolol is kind of formulation specific, which was done by numerical deconvolution. So whenever we are doing some different formulation, we might have to look critically that how it might perform well when we are using the in vitro dissolution input. And third thing is that this conceptual framework, it can be also applied to predict the outcome of a new drug formulation after linking the dissolution kinetics with developed PBPK model. So it's not only actually restricted to metoprolol. We are also working with BCS class 2 and class 3 compounds where we will be using that particular in vitro dissolution as an input, and then we'll be checking the performance of the model. So just future direction, I would like to mention that for healthy volunteers, we did this study for the healthy volunteers, but PBPK model has the unique opportunity because there you can actually also vary the system parameters and as well as the drug parameters. So after changing the trial design of the population system, you can also do perform a in silico B evaluation, and just to determine that how your B parameters are changing, or like whether you are getting VIN or not. So with that, I would like to wrap up my presentation, just to acknowledging my mentors, Dr. Lesko and Dr. Smith, who actually helped me to identify this project and and also gave the opportunity to use this particular framework. And also, I would like to thank my co-worker Haita, who also did who used DDDPlus to give me a good dissolution input and output to determine the absorption profiling. And also, I'd like to thank our collaborators, FD collaborators, who have given a critical input when we are doing this particular project. And also, I'd like to thank Simulation Plus because of this particular component tools we can actually use to address different type of other clinical pharmacological questions as well. So with that, I'm ready to take any kind of question.
Okay, Sumit, thank you very much. We've got some questions come through online, and I'm going to start with the ones for Jim initially. So, Jim, let me have a look at these questions and see which ones we can cover. So there's one here. Is it possible to convert the in vivo release profile to in vitro within GastroPlus, or should we do it manually using the equation you showed?
I mean, recently in GastroPlus, we added a tool that allows you to input your in vitro data, and it will automatically use your IV IVC data file and calculate in vivo dissolution. However, going the other way is a little bit more, you know, it's not currently, I think, automated. So you would have to go the inverse. You would have to, you know, solve that yourself. But that's a good idea for a new feature to add to the software.
Okay. And then another one which asks, are the calibration constants for excipients formulation independent, i.e. pertinent to the excipient only regardless of the formulation approach?
So if you keep the same sort of tablet formulation platform where you have the same excipients and you're just making some adjustments of those excipients, then the parameters can translate. If you go to adding completely new excipients, completely changing the system of your tablet, that's the parameters are not going to be transferable most likely. So that they're going to be useful in sort of the platform technology that you're using for that specific formulation. But
yeah, thank you. And then another one, how do we estimate the relationship between in vitro variability and in vivo variability?
So in vitro variability is strictly we're strictly looking at the in vitro variability of the in vitro experiment in the case that I was showing. The in vivo variability is taken into account based on the population simulator within GastroPlus. So that puts variability on all the, volumes and pHs and et cetera, et cetera. So the population variability is determined by your population model setup in GastroPlus. Now, when you use the when you bring those in vitro profiles over to GastroPlus, you could actually add variability to those high and low profiles if you want. But I think the goal there is that we already are picking out the extremes of what we expect the dissolution variability to be. So, I mean, I don't think you need to add more variability on top of that. Once you bring it into the population simulator, you can just simply use the variability of the subjects then using the population simulator to dictate what your in vivo variability is. But yeah, those are interesting, good questions.
Okay, and in the case of developing a generic formulation for innovator product where such IV IVC data is not available, how do you suggest to use DDDPlus and GastroPlus modeling? So generic formulation where we don't have IV IVC, is it necessary to use data from failed bioequivalent studies in humans?
I mean, this approach can be undertaken with as little as one data set, as long as you have one data set. Now, how predictive is that going to be if you make large changes in formulation? It's probably, your mileage is going to vary, right? But I mean, sort of the implied, thing behind this whole method is that you're going to have one formulation so that you understand the in vitro dissolution and how that converts to the in vivo dissolution. If you don't know that, this method is going to have maybe some limited applicability. What you're going to have to assume is that your in vitro dissolution experiment is one-to-one correlating with the in vivo dissolution rate, which is not always the case. I mean, for CR formulations, it's going to be probably closer to true than other formulations like IR formulations, but still, that's kind of the whole crux of the method here, is that you can't make an in vivo prediction unless you know that translation from in vitro to in vivo. So at least one data set, and you could maybe take this from a failed bioequivalent study, or maybe if you can find literature data for a given formulation and you can find in vitro data and whatnot, so there's other ways to try to get the data if you don't have the study, maybe try to use the literature. But
yeah, just as a follow-up to that, Jim, when you were presenting one of the examples with the fast and the slightly fast formulation, I noticed that you use the slightly fast to predict the fast. So you're essentially extrapolating outside of your data set rather than interpolating within.
Yep.
Do you have any sort of comments on that?
My comment would be, yeah, this is sort of an example. My comment would be is what you would want to do if you were doing this and you wanted to, sort of minimize what you're, the experiments you're doing. What I would do is do the slowest and the fastest and try to get data on those. And then that'll allow you to interpolate, as you said. So you are correct. That is the ideal way to do it, for sure.
Yeah, great. And then there's a question here. Can you let us know how DDDPlus can help in case we have a USP4 dissolution data only for IM products? So I'm guessing that's intramuscular.
Yeah. We currently have, there is a USP4 model within DDDPlus, so you could model as an IR, you would call it an IR powder in DDDPlus, but you could add, maybe and do some dissolution experiments in DDDPlus for that. Now we have added some new features in terms of IV IVC with intramuscular. So I mean, these are going to be coming out. Some of these new features are going to be coming out in the next version here in a few months. So we're going to add some increased functionality within GastroPlus to deal with some of the long-acting injectables. We also have a model for long-acting injectable formulations that we're adding to DDDPlus based on an FDA grant that we've been working on. So there'll be the possibility of using that these are for PLGA type formulations. So there is a couple things that can be done, and there is some more. There is some new things coming down the pike, I guess you could say that for that.
Just a thought again on this USP4. So if DDDPlus can or has a facility to look at USP4, can we sort of train the software in using USP2 data and then predict what the dissolution profile would look like in the USP4?
Yeah, I mean, if you have a model that's built for USP2, theoretically, the solubilities, the release rates and everything. Theoretically, you could probably move that to the USP4, and based on the flows and the concentration gradients that are produced in that experiment, we can calculate that. Presumably, if the differences in release are just based on those concentration gradients and differences maybe in the fluid velocity in the two experiments, I mean, theoretically, the model should, help predict that when you translate.
Great. Okay. So there's just a couple of questions here relating to clinical endpoint. Is that sort of within scope of this, or is that a different topic, do you think? How can we predict clinical endpoint using the modeling? What factors to consider?
I mean, yeah, I don't know if that's, part of, I haven't really thought that much about that in terms of this method. And you mean sort of how to adjust our clinical studies to decide when the endpoint is, or?
Well, I think the question is referring to how can you relate in vitro, in vivo dissolution data with clinical endpoint. I think that's probably a step on from what you were describing. Actually, you probably need more clinical data to do that.
Yeah, I would agree with that.
Yeah. So I'll just see if there's any questions for Sumit. Okay. Yeah, Sumit, I'm just looking to see if any of these, I'm trying to balance it across the two presentations, so just seeing if any of these are relative to yours. I do have one question actually for Sumit, particularly when we're talking about extended release formulations. To what extent do you think the selection of the method in your dissolution methods, so in terms of method parameters, the shear perhaps in the system, might affect your ability to predict for different extended release formulations, or particularly for extended release formulations, which are dependent on the response of the polymer to shear, for example?
Can you hear me now?
Yeah, we can hear you now.
Yeah. So I can answer with this with the dissolution profile simulation. Like for DDDPlus, for my experience, BCS+1, you don't have to worry about the dissolution input parameter of that particular drug. But whenever you are working with the BCS+2 or BCS+4, then you have to add the other mechanisms like disintegration profile maybe, or the precipitation kinetics as Jim showed there. For extended release formulation, I can think about for BCS+2, not only solubility, sometimes the slow dissolution rate can predict like less than or bioinequivalence for the generic drugs. So in that perspective, so I think we should first develop the DDDPlus model for that particular drug, and then test in different medium using different pH or also the release rate. And after that, we can actually connect with the PK profile to determine how off we are with the observed data. So that might be one factor. And second thing, apart from sometimes what happened for the BCS+4, we have both low solubility and low permeability. So we cannot integrate that particular factors. But at least if we know through DDDPlus that what are the dissolution profiles are there, we can like kind of strip out that particular phenomena by using DDDPlus and then linking with the GastroPlus, just by like covering off the dissolution input and just say that how poor absorption is there because of the low permeability.
Okay, thank you. So just time for just a couple more questions. How does the software predict mouse, rat, dog, and monkey PK and TK? So I don't know, Jim or Sumit, whether you've any experience in terms of, you know, within the context of this webinar, sort of predicting dissolution in animal species.
No, I think it's not dependent on the species. It depends on the API. For DDDPlus, just you have to put that particular API, the chemistry and the manufacturing condition, and then only you can get the dissolution input. For the monkey, rat, or dog data, you have to just do the drug, I would say GastroPlus model. So in the animal model, you have your absorption profile and the disposition profile, so you can keep constant for that particular animal model, but the dissolution profile will be same for every API.
Okay, thank you. And when it comes to, I think this was in Jim's presentation, the overestimation of precipitation, what do we think are the factors that are contributing towards that overestimation of precipitation that you noted? You said in vitro, typically we overestimate. Do we know why?
I think our theory and why we're moving towards looking at some of these other in vitro experiments, like the ASD model, is that we don't have, you know, a realistic in vivo situation where we have, you know, the dilution in different media, you know, as the drug transits from, you know, the stomach to the intestinal media, along with you know, the precipitation going on. So we've seen that there's been in the literature just recently, and some of the work that's been going on in the Orbido project, that it seems like using data from the ASD where you have more realistic conditions as far as, you know, more similar to the intestines as far as the fluid volumes and compositions of the media. It seems like these are providing inputs that are more realistic as far as the in vitro or in vivo simulations. So I think that's where we're trying to get to as far as building the new models. And I think the closer you can get to the realistic in vivo, you know, conditions, the better the precipitation parameters that you can extract from these experiments. So I think that's what you know, is sort of coming out of all this data, is that maybe some of these experiments where there's extraction of the drug and or more realistic fluid volumes and buffer conditions are what's needed to really improve these predictions. So
Great, thank you. One last question is asking, do you have an absorption model available for intra-articular injection?
I don't think we have that.
No, this is Vera. I'm sorry. No, we don't have that administration route currently as a specific route. So
maybe one to put on the list. Okay, so I'm going to finish the questions there. I think we've covered most of them. And I'm going to finish by saying a very big thank you to both Jim and to Sumit for their very interesting presentations, and also to the listeners, and particularly those who've put forward questions to help with the discussion at the end. So thank you, everybody, presenters and participants, for your for joining today and for a very interesting session looking at dissolution, DDDPlus, and how we can use that in IV IVC. So thank you, everybody. As I say, this webinar has been recorded, so those who want to catch up with it, it will be on the website normally within a week or two, so keep a lookout for that and for future webinars. And thank you very much. Goodbye.
Bye. Thank you.
Thank you.